Hi, I'm Dr. Arsalan and today we are going to explain the third component of aerobic respiration, the electron transport chain. It may be defined as it is a series of protein complexes and other molecules which transfer electrons from donors to acceptors by redox reactions and release energy from NADH and FADH2 molecules obtained in the Krebs cycle. During the Krebs cycle, the NADH and FADH2 molecules were produced. Basically, these are the coenzymes. NADH and FADH2, these are coenzymes, belong to the class nucleic acids. In these molecules, huge amount of energy is stored. NADH refers to the nicotinamide adenine dinucleotide. This is its reduced form. And the second one is reduced flamin adenine dinucleotide. These two molecules were obtained during the Krebs cycle in our previous topic. Electron transport chain, basically it is aimed to release the stored energy in the NADH and FADH2 molecules obtained from the Krebs cycle, because Krebs cycle has produced the energy in the form of these coenzymes, which is indirect and cannot be directly used by the body. Firstly, these should be converted into ATP molecules. So for the conversion or release of ATP molecules from these molecules, the process is called electron transport chain. Explain the definition. So it's a series of protein complexes. We know that electron transport chain has four complexes which are written in Roman numerals 1, 2, 3 and 4. And other molecules, these molecules are cytochromes. So electron transport chain or electron transport system, it comprises four complexes along with two cytochrome molecules. These molecules and complexes are aimed to transfer the electrons from donors to acceptors. The donors are NADH and FADH2, while the acceptor is oxygen molecule. When the electrons are being transferred from these donors to acceptors, the process is called redox reaction. Redox reaction means during this process, simultaneously both reduction as well as oxidation reaction are taking place. Redox means reduction as well as oxidation. So its short form is redox. During this redox reaction, when the electrons are obtained by the electron carrier, this constitutes reduction reaction. And when again these electrons are delivered to the next carrier, the same carrier is being oxidized. So at each step, oxidation and reduction takes place, which releases huge amount of energy, which is coupled by ADP and phosphate group to synthesize ATP molecules. So the electron transport chain, this is basically aimed to release the stored energy from these coenzyme molecules and form ATP, which is directly or readily used by the body. The electron transport chain, just like Krebs cycle, it occurs in the mitochondria. The energy is released in the mitochondria, therefore mitochondria is also called the powerhouse of the cell because it generates ATPs and energy from the glucose molecules. There are six components of electron transport system or electron transport chain. These six components has four complexes and two other molecules. The complexes are mentioned in Roman numerals: complex 1, complex 2, complex 3 and complex 4. The complex 1 is NADH dehydrogenase. Basically the complexes, these are set of enzymes. These set of enzymes are called complexes. The first complex was NADH dehydrogenase. Second complex is FADH dehydrogenase. Third complex is cytochrome reductase. And fourth complex is cytochrome oxidase. The fifth component is coenzyme Q and sixth one is cytochrome C. These are the six components of electron transport chain. As we know that during Krebs cycle, one glucose molecule in Krebs cycle, one glucose molecule yielded 6 NADH molecules and 2 FADH2 molecules. These two molecules are coenzymes These contain huge amount of energy stored in them, and the electron transport chain, basically the function of electron transport chain is to release the stored energy from these molecules. As these molecules contain hydrogen, therefore we can say that these are in reduced form. The electron transport chain, these molecules lose electrons, and when electrons are lost, this is called oxidation. So basically, electron transport system oxidizes these two reduced molecules. These electrons are delivered to the electrons carriers, and then final acceptor, the oxygen molecule. So these are donors, and oxygen molecule serve as acceptor. This is the final acceptor. The electrons flowing from these donors towards the acceptors, at each step the reduction and oxidation reaction takes place, releasing huge amount of energy. And this energy is utilized during the process of formation of ATPs. The ADP molecule combines the phosphate in the presence of this energy and produces ATP molecule. So this is the basic mechanism of electron transport chain. The electron transport chain is common in both respiration as well as photosynthesis. The mechanism of electron transport chain is very simple. It involves all these six components in its mechanism. Let's see here. The first molecule, NADH, it reacts with the complex 1. As we know that complex 1 consists of NADH dehydrogenase. Similarly, the FADH2 molecule reacts with the complex 2, which comprised FADH2 dehydrogenase. Both these molecules, when react with their relevant complexes, electrons are released from these molecules. This is the symbolic representation of electrons. When the electrons are released from both NADH and FADH2 molecules, these electrons are handed over to coenzyme Q. When the electrons pass from these donors to the coenzyme Q, these donors are oxidized due to loss of electrons, and this coenzyme Q is reduced due to gain of electrons. Again, this coenzyme Q delivers these electrons to the onward complex 3, the cytochrome reductase complex. So when coenzyme Q lose electrons, again it is oxidized, and subsequently complex 3 is reduced due to gain of electrons. So this constitute redox reaction, reduction and oxidation reaction. Due to this redox reaction, energy is released. So at each step, energy is released due to redox reaction. This energy is accumulated and it is utilized in the synthesis of ATPs. Finally, these electrons moving from the donors to the coenzyme Q, then complex 3, the cytochrome reductase, then cytochrome C, then complex 4, cytochrome oxidase, are finally delivered to oxygen. Oxygen is the final acceptor of electrons. This oxygen molecule, it obtains electrons and hydrogen inside the matrix of mitochondria and form water molecules. So the water molecules formed during the process of respiration comes from this electron transport chain mechanism. It should also be kept in mind that during this process of flow of electrons, the hydrogen ions which are released from these coenzymes, these molecules, the hydrogen ions produced in the inner matrix are pumped into the intermembrane space. This is the diagrammatic sketch of mitochondria. The outer cover or outer line indicates the outer membrane. This one is inner membrane having bulky matrix in it and enzymatic components as well as the components of electron transport system. The space between the outer membrane and inner membrane, this is called intermembrane space. The space lined by the red lines is the intermembrane space. So accompanying the flow of electrons during this transport mechanism, the hydrogen ions liberated from this FADH2 and NADH are pumped from the inner membrane to the intermembrane space. So the concentration of hydrogen ions subsequently increases in the intermembrane space, the space between two membranes, the first membrane and the second membrane. The space here, it is called intermembrane space. Intermembrane space is accumulated with the hydrogen ions. Hydrogen ion concentration is increased in the intermembrane space compared to the inner membrane or matrix. So the concentration gradient is created. This is called concentration gradient. And we know that the ions or molecules flow from high concentration to the low concentration. So the hydrogen ions will move from high concentration of intermembrane space to the low concentration of inner mitochondrial matrix. This movement will be called as osmosis because they are crossing the barrier of membrane. So the movement of hydrogen ions from the high concentration to the lower concentration across this mitochondrial membrane, this is called osmosis. As these chemicals are moving through the process of osmosis, so this will constitute another mechanism of chemiosmosis. Chemiosmotic ATP synthesis, its literal meaning as well as definition reveals that it is the combination of chemicals and osmotic processes. So it is the process in which ATPs are synthesized by the combined chemical and osmosis processes. As we had explained in this mechanism, that the hydrogen ions being chemicals flowing from the high concentration to the lower concentration constituting osmosis releases energy. This energy will be utilized in the synthesis of ATPs. So ATP synthesis occur due to the osmosis of these chemicals constituting chemiosmotic ATP synthesis or the process in which chemicals by the process of osmosis liberates energy which is utilized in the ATP synthesis. The chemicals liberating energy due to osmosis utilized in ATP synthesis. This process is called chemiosmotic ATP synthesis. For chemiosmotic ATP synthesis or the synthesis of ATP, an enzyme ATP synthase is required. This enzyme ATP synthase, it is activated due to concentration gradient. The concentration gradient is achieved, the enzyme ATP synthase is activated. ATP synthase has two components. The first one F0 and second is F1. These are also called oxysomes or elementary particles and are present in the membranes of mitochondria. The F0 particles, these are present in the membrane of mitochondria, inner membrane of mitochondria, and F1 particle is also present in the inner membrane of mitochondria projected towards the matrix. The F0 particle, it is related to the flow of hydrogen ions from intermembrane space towards the matrix. Flow of hydrogen ions from the intermembrane space towards the matrix, it is accompanied by the F0 component of ATP synthesis, and the F1 particles, these are required for synthesis of ATPs from ADP molecule combined with the phosphate using the energy liberated during this chemiosmotic process synthesizing ATP. So this process is catalyzed by this F1 particle or elementary particle or the component of ATP synthase. So in this way, the NADH and FADH2 molecules produced during the Krebs cycle are oxidized in the electron transport chain and releases energy in the form of ATPs. It should be kept in mind that one NADH molecule releases 3 ATPs and one FADH2 molecule releases 2 ATPs. So we know that during Krebs cycle 6 NADH molecules are produced and 2 FADH2 molecules are produced. So if we multiply 6 by 3, it gives 18 ATPs and FADH2 gives 4 ATPs. So these ATPs are released during the electron transport chain which were stored in the NADH and FADH2 molecules. So it can be concluded that Krebs cycle no doubt produced huge amount of energy, but the energy was in indirect way in the stored form in these coenzyme molecules. The body was not able to use these molecules directly as a source of energy. So the basic contribution of synthesis of ATPs or energy currency for the body is of electron transport chain because it utilizes these coenzyme molecules along with the set of chemicals, enzymes and components of the electron transport system in the mitochondria. These ATPs from these coenzymes Now these ATPs are readily available to the body and can directly be utilized for other activities of the body and serve as direct source of energy. So this was all about the aerobic respiration, having three steps. The first one glycolysis, the second Krebs cycle, and the last one electron transport chain, also called electron transport system.